A very good morning to all of you. I, Dr. Priyanka Kriplani, on behalf of Organizing Committee, welcome you all to Guru Gobind Singh College of Pharmacy, Yamunanagar, on the third day of the 6 days AICTE-ISTE sponsored induction program held with the theme, How to Plan Experimental Methodology in Pharmaceutical Research. It is our privilege to welcome all the dignitaries of the today's session, Professor O.P. Katare, UIPS, UGC Center of Advanced, Punjab University, Chandigarh. Dr. Rahul K. Verma, Scientist, Institute of Nanoscience and Technology, Mohali. Dr. Sanjeev Kumar Manmundar, Manager, Research Development Corporation, Delhi. Dr. Kumar Gaurav, Principal, Guru Gobind Singh College of Pharmacy, Yamunanagar. I welcome all of you. I also extend a heartfelt welcome to all the participants. So for today's session, first session, Professor O.P. Katare, UIPS, Punjab University, Chandigarh, he is the first speaker of our day. He is well known, widely acclaimed, world's top-rated scientist of Punjab University, Chandigarh, and is known for his multi-dynamic persona. There is a long list of achievements and accomplishments. He is recipient of National Technology Award by President of India, late APJ Abdul Kalam Sahib, in 2007 for industry research in the field of nanoliposome technology to benefit psoriasis in affected patients directly, that brought him to the international repute. Only few days back, a prestigious pharma company, IPCA Labs Mumbai, has launched a novel nanotechnology-based Itova LP gel to help relieve pain and inflammation. Besides several patents, products, and publications, he has contributed immensely towards the pool of human research-ready resources by guiding more than 2 dozens of PhDs. And further, on the other point side, he takes keen interest in youth empowerment activities by conducting regular workshops, seminars, and certificate courses on the personality development with special focus on heartfulness meditation practices. I welcome you, sir. I welcome you, sir. So can you hear us? So we can't hear you.
Hello, am I visible?
Hello.
Yes,
Yes,
sir,
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 we can
you
hear
are
you.
audible and visible.
Can you hear me?
Yes, sir,
Yes,
yes,
sir. Yes, sir.
Okay, there was some problem in the mic, I think. Now it's okay.
Yes, sir. Yes, sir.
Is it okay now?
Yes, sir.
Okay. There was some problem in this mic. Speaker.
Yes, sir. Yes, sir.
Is it okay now?
Yes, sir.
Okay.
हेलो
हाँ अश्विन,
Good morning, sir.
गुड मॉर्निंग। किसी का वेट कर रहे हैं क्या?
मैं एक मिनट से चेक करता हूँ।
Because everything
मैं
is
बताता हूँ।
it seems totally silent. And what is the gap? I'm waiting and waiting.
मैं एक मिनट दीजिए सर, अभी बताऊँ।
Sir, I request you to please take over. Sir, can you hear me?
Yes, I hear. Where is Dr. Gaurav? It is muted.
Very good morning, sir, and welcome, sir.
Thank you so much. I was to hear your voice before I start.
Okay.
That I was looking for an actuator, like in an aerosol, you actuate something. So it's a, I was looking for that.
Okay, sir.
And I'm not actually finding myself in the audience. It seems. Is it so, Nidhi? क्या नाम है आपका? Am I available?
Yes, sir, you are audible and please take over, sir. We are waiting to hear you, sir.
Acha, how much time is given?
As much you want, sir.
Oh, I have 1 hour only. Yeah. No.
Okay, sir.
There are so many things rushing around.
Definitely, sir, we can understand that.
Nowadays, everyone is in whirlpool of the activities. Okay, I share my screen.
Okay, sir, please. Please, sir.
So my screen is visible now?
Yes, sir.
Okay. So let me once again, my hearty welcome to my hearty greetings to you all the participants. I hope all are teachers and some research scholars.
Yes, sir.
Okay. So pharmacy has a great future, especially in the corona time. Now the society, the government, everyone is looking for pharma with a great interest and hope. दवाई का अभी जितना इम्पोर्टेंस अभी समझ में आया है ना, उतना पहले कभी नहीं आया. आई होप एवरीबडी विल एग्री. So this presentation actually I have designed to give some of my what we call thoughts, feelings, and the distilled whatever you may name it as a wisdom out of it, कि अभी हमको करना कैसे चाहिए. रिसर्च प्लानिंग ये वो बहुत सारी चीजें हैं, बट द केस टू केस इट डिफर्स. So I am sharing my own, how did I plan and how did I move forward, and now at this juncture what is where we stand. तो हमारी जो जर्नी है वो हम आपके साथ शेयर करने वाले हैं. इसका जो टाइटल मैंने रखा है वो ए वे फॉरवर्ड फॉर ट्रांसलेशनल रिसर्च एंड बियॉन्ड. That attracts the attention that we have lot of science being done, lot of science being published, even patented, but until unless we convert it into a real-time solution product and something which is worthwhile to be used by the society, it is not a success. So sharing my story and the technology and what I think where we stand as of now, this presentation is all about that. So the first slide is the Gandhi Bhavan. This is our iconic building at Punjab University campus, where we actually get lots of inspiration how to excel in your life and what sort of a spiritual spirit that the higher spirit the higher human spirit one should actually inculcate, acquire. So saluting this, the iconic, this symbolic building of Gandhian spirit. What I am to share, which is not introduced about me in the formal introduction, that I hail from a very humble background. All my training, all my education, the primary education was done in the very a village school. And mind it, it is all about this village school education. Whatever I have got today, where I am today, it is actually, it goes to the credit goes to the those primary school where I learned the human values, the religious background. And though my father was a professor in agriculture college, one place, Riva in MP, but he left me with my grandfather, who took care and and actually gave me all the sanskars and all that, which is, and those Ramcharit Manas. So heartfulness, which I uh, give the final credit to my so-called success. It may not be success, but at this point of time, whatever it is there. So whatever I got, it is because of the background, the very humble background, and that रामचरित मानस की वो चौपाइयाँ जो होती हैं वो अभी भी मेरे कानों में गूंजती हैं. उसमें से एक चौपाई है जो मैं आपसे बाद में शेयर करूंगा. बट I take pride actually in getting the education, and the very where I lost all my English, Angreji, and I still regret not being not being able to get through those English school and that. But what I got is much beyond that. So this is the focus, and I will talk a little bit of technology, but and the story, and then the आज की डेट में हम क्या सोचते हैं, हमको कहाँ जाना चाहिए, कैसे करना चाहिए, बहुत बड़े प्रश्न है हमारे सामने. अभी हम देखते हैं कि lot much of mushrooming of the pharmacy institutions. इतने सारे हर गली नुक्कड़ पर एक pharmacy institute. In our time there was only few institutions across the country, and it was very difficult to get the education in that, like BHU and Sagar, where I am groomed. बहुत कठिन था. और अभी हम देखते हैं कि there is a lot of dilution, and you can easily get the seat, you can easily get the permission of pharmacy institute, and quality standards are so down. तो इसके चलते बहुत कुछ जो हमारे expectations हैं वो हम कैसे meet करें, इस सब के बारे में कुछ मैं सोचता हूँ शेयर करना चाहिए. So philosophy, it always anything starts, the knowledge starts from philosophy only. महात्मा गांधी याद आते हैं हमको जब भी हम बात शुरू करते हैं मॉडर्न मेडिसिन में, फार्मेसी में. वो कहते थे कि मैं अंग्रेजी दवाई नहीं लूंगा. Why? Because he knew that all this chemistry, the chemical molecules, they are not safe, and safety defeats the efficacy, and even they are not effective, and they are not very compliant. So there is no way out for us. We have to challenge Mahatma Gandhi. Actually, it's not like challenging the Mahatma Gandhi, but challenge we have to accept the challenge that we want the medicines which are really effective and while safe and compliant, and only research through innovation we can evolve, we can progress. So research is very, very important in these times of viral times, where the viruses rule the world, and we are looking for the medicines. So research is not an option but a compulsion. So how to acquire that skill, that attitude, that aptitude, that's very, very important for us. Let's so this is a very popular that jump, bump, and dump model, where this is when we take a drug, it is just like putting inside the biological system where the molecule jumped and bumped and dumped wherever they like to be, according to their own making chemistry, physical chemistry, and according to the biology. There is no guidance, the guided principles. So on the face of this current situation, we are looking for an ideal situation. We can actually make the magic bullets uh, and can hit onto the specific targets, specific affected site. So the future will belong to the targeted delivery system, and we have been working for it. And so actually we need a very uh, focused kind of a delivery where we look for the technology. So liposome, now in the times of the amphotericin B, liposome is not a new thing. These are very versatile kind of a host carriers delivery structures ranging from micro to nano, and they can actually they are based on a very unique uh, polar lipid class 2 polar compound. They're called phospholipids, and because of their uniqueness in their structure, their chemistry, they can easily get into the self assemblies when they come into the contact with the water. So water is the deriving force which lead to formation of these structures, vesicles, closed laminar vesicles, because of their thermodynamically driven perturbations and leading to the stability, energetically favorable state. So their diversity uh, attracts the scientists, the delivery and formulation people. Then they have lot many things to offer, ranging from the solubility, their targeting this ligands, and their uh, so much of the meritorious features they had, and specifically the ability to entrap wide variety of molecules. So now we have the vaccine, which is on the same principle, same platform, similar platform, where phospholipid played a wonderful role to deliver that epitope to create that vaccine. They can be structure differently depending on the type of the lipids or the type of material, the combination, the conditions, the process. Varying all those factors, it can produce the uh, typical structure to suit the particular molecule to be entrapped. So there can be a kind of a tailoring, putting up a tire to the molecule according to the need, according to the application. So we have enormity of the possibilities to create the variety. So they are having the different parameters. And their features are different. Unlike the conventional system, they are to be standardized. The quality parameters are different. Like, for example, the micromeritics, the lamellarity, the phase behavior, phase transition phenomena. It plays a role in liposomes targeting, particularly where they can release the drug depending on the stimulus, temperature, pH, or other signals they can use. So they offer the opportunity to be designed according to the need, medical need. So these examples encourage us that there is a proof of the concept. This amphotericin B and doxorubicin, launched by even by Sun Pharma, and recently Marcubo. This is a vincristine. These examples actually make us understand that it is possible, and they are the one which are most explored in the history of drug delivery systems and targeting because of their bio-friendly compatibility and not creating the nuisance like particles and nanoparticles and all and other polymeric systems, and they are always a chance, a risk, but these systems are quite acceptable to the biology of human biology. So coming to the second part where I want to share my story, that how did it happen, the translation at the University Institute of Pharmaceutical Sciences. We are number, actually we are ahead of any institution, but NIRF ranked us number 2 for last, since its ranking begin, we are at number 2. We are standing at number 2. And the Professor Harkishan Singh, who doesn't know the name, late Padma Shri, the only Padma Shri that decorate, that embellish the department, Punjab University. The only molecule given to the country is coming from the Punjab University UIPS, thanks to the Professor Harkishan Singh, the father figure. So UIPS has the long history of excellence and the record. Many stalwarts like Professor Kulkarni, Professor Handa, and Professor Chopra, in the history, in the Indian pharmacy history, the glory of UIPS is unparalleled, unmatched. So when I joined the department 1997, sorry, and 30th April, I still remember, I was encouraged by the example that, thank God, was launched by the Palicia Biotech, through where we had collaboration. Professor Singla was instrumental to put this to get this research out. So in this very inspirational background of the UIPS, lot of motivation. So basically, how do we plan in pharmaceutics and drug delivery? You have to choose a drug, you have to think of a disease, and you have to think of a formulation that you are working on. So I had all these things coming to me. And the very first project that I took for the PhD converted into the product. And since then, there is a lot of research done, and still it goes on. But this is the standard, gold standard story, which still encourages me and my team, and it has its own thrill. इसका एक अपना अलग ही कुछ वो है, there is a charm in it. So what happened actually, this disease was very difficult to treat, and psoriasis, everybody knows now, and there worldwide the population affected, number of organizations, NGOs, they're working, and even one organization is for all the organizations, that is a very difficult and a very rare observation with any disease. So actually, what happens, there is a overproduction of the skin cells, which creates the difficult pathology, and this is actually immunogenetic in nature. So there's no good medicine. Systemic therapy and topical therapy both are having their own deficiency in gaps. You just think of a situation where you are using methotrexate systemically and cyclosporin to suppress the immune system. How hazardous it is, and then topical therapy is also not very good so far in this scenario. So both approaches, medical approaches, they are not actually very satisfactory. So we started working on this molecule diethanol, which was actually being suggested by doctor, dermatologist at PGI, Professor Bhushan Kumar, who actually was the mentor of this project, who gave the idea that why don't you work on this molecule, which is very good but not friendly, but it burns the skin, it stains the skin. So we need a good product of this drug, which is not available in the market. The Darowin is really not acceptable. So it was banned twice, and then again it was brought back to the market because the drug was so good. So it is a typical case when you want to use it, but you are not very happy to use it. So it's a very paradoxical situation where the drug is good, but drug is not good. So many papers and so many patents, so many, nothing worked actually. And its beauty is to in it is to hit the multiple targets such located at the cellular, intracellular, and intracellular levels, but its bad effects overweighing the good effects. So in this situation, it is a fearful drug. And people started working, researchers, they tried to tamper with the molecule to create different derivatives, but you can see that there is a improvement in the staining and irritancy, but at the same time efficacy is also down, because this efficacy is related to its ability to produce some staining and irritancy. So there is a paradoxical situation कि आपकी जो एफिकेसी है वो आपकी उस बेड क्वालिटी के ऊपर डिपेंडेंट है. सो this chemistry, this chemical approach could not work, and whenever they tried to change the molecule, and there it did not work. So people tried differently using different vehicles, nothing worked. Clinical modality and miscellaneous methods, nothing worked with this molecule. This was the feedback from the PGI for this Dharma head, and he said nothing is working, Professor Bhushan Kumar. So this was our observation only, that we could dig out what is the problem. Actually, it has got this auto oxidation ketoenol tautomerism, where we have the degradation leading to first generation, second generation, and third generation. And actually, it is the middle one which is required, but the degradation is so fast that it turns out to be a next generation products. So because of this mechanism of degradation, the drug is needed in a higher dose and irritant outcome. So we started working on it. Interestingly, there was a German researcher who wrote in Gisperation in the one journal, 1992, Mustakelio, that it is very difficult whether an entirely non-irritating diethanol derivative treatment modality will ever be developed, because he just handed down or surrendered कि इसका तो कुछ हो नहीं सकता है क्योंकि इसमें ये प्रॉब्लम है. तो उस समय उनको ये आइडिया नहीं था कि हम कौन सा फॉर्मुलेशन, कौन सा ड्रग डिलीवरी, क्या इसको कर सकते हैं. So this is how the idea cropped up in the classroom teaching situation, where I was teaching a simple physical pharmacy to use phospholipid lecithin in differently to tame down its modify its reactivity. So we used it, and this is a real-time picture of a liposomal system where the liposomes could entrap the diethanol molecules, and because of this, the reactivity is modified in a, so trend is reversed when liposomes could actually took the process upward rather than downward. So we are getting more and more anthonyl radicals here. So we got the design in the formulation to fetch the patent. We got multiple worldwide patents, and we had a clinical trial where we could demonstrate that only 0.5% is equivalent to 1.15%. So there was a thrill when the resident doctor at PGI reported that the product is working excellent, and that excellence, that excellent word, the sound is still echoes in the ears, in my ears, which keeps me going, fueling my interest, my enthusiasm till today. So then the company came into the picture, hearing the story that the doctors are approving the product in the clinical trial, and they are happy with it. So we had a technology transfer. Our vice chancellor and all our luminaries, they were so happy to have this tech transfer. The Professor Bhushan Kumar, the dermatology head, actually it is the proof that how clinical insight is important. we remain aloof of the clinical insight, and we do not get along with the clinicians, or they do not come to us. That is the one problem which is facing. So research plan is always in association with the actually the person with the clinical insight, what actually it is needed. Then we had these all our registrar and the vice chancellor, Professor Pathak was there. Then making a drug product in the lab is a different thing, and scaling up is a different thing, and particularly in the system where you have to control so many parameters, vulnerable parameters like entrapment and all. It is not easy a task to scale it up, but we did it all these with the help of the team, and all these the design of experiments our professor group helped, and in stability Dr. Sanjeev from Naiper. So I took help from all the people associated, the best one in the region, and they were very heartful and they were very willing to support my research, whatever be the reason. I do not know, but they uh, actually was very, very kind to me, and I'm thankful to all those people who made it possible. This turned out to be the reality. That life care has brought into the market on the name of Surizone. Marketing muscles are not very powerful, so it is not been so widely available. Yet we are struggling, and we are trying to get it with the marketing muscles, looking for the partners all around. Hopefully we'll do it. And this is how the company market, from friendly to from fearful to friendly formulation. And then we went on with this translational work with the many other concept. One is the coal tar, using different approach with the nano formulation. And then we had we have been working with the in association with the microbiologists, putting all these bacteriophages, the virus, into the liposomes to overcome the biofilm and the antibacterial resistance. This is one example where we could fetch the patent, two patents. One is under the PCT application with the ICMR support. And then we had, we have been working on, and this is PGI is a actually boon for us, that across the road we are able to collaborate with them, and academically collaborated research projects. So we have all these opportunities of working, and we are working for other drugs like methotrexate or Delavine for the rheumatoid arthritis. We have also been working for some vaccines. We one of my students, Dr. Anshuman, he worked for this biolosomes, and we got the patent using these system through oral, and which gives the comprehensive results. And so all these examples which reflect on the outcome of the research, and many multiple projects are in at the verge of converting into the real-time solution for different applications. So we have a collaborative model. One cannot work alone. One has to collaborate and network. And this is what new education policy also talks about: key collaborate, collaborate, and collaborate. But how to collaborate is a difficult million-dollar question. It needs a lot of thinking and a lot of input, and so different dimension. You have the knowledge, you have the expertise, but even then you are not able to collaborate, get along with the people. So, but it is required that you have to have industry with you, you have to have the medical institutions with you, you have to have your inner circle very cohesive, very strong team inside the institute. So all these factors influence the outcome in the academia industry partnership, where need a support from everywhere. So this product turns out to be a major success when it could fetch the award, national award, with President of India. Or I had a fortune to shake the divine hands with the Kalam Sahab. Not only I got a shake hand, but I also got a lifafa of 1 lakh rupees, which was on those times was a huge money. But it's a very handsome reward for this. And we had further on, we could I could OPPI scientist here also. Another envelope of 1 lakh rupees from the Andrew the GSK chief at Hotel Taj Mumbai. So बहुत कुछ मिला, बहुत कुछ रिकॉग्निशन मिला मुझे भी, इंस्टीट्यूट को भी और यूनिवर्सिटी को भी. Now it's a happy to share that our students are doing wonderful. This is Dr. K. Saraya winning the US award for QBD oriented systemic approach. So it is a more of a happiness when you see your students are being recognized and they are ready to do even better than you. It's a real return and reward of your work when you see your students rising and rising. They are coming under 2% scientists and all that. So we had a lot of hits in the headlines of the newspapers. So it kept on, and there was awards were raining, and wherever we applied, we got the award. And this is the recent the Jaipur APTI Scientist Award. And I was made director research of the institution because of my translational success. I'm not that intelligent and that competent as vis-a-vis other on the campus, but because of this conversion, this translation of the academic idea into the industry product, it was valued, and I got this position of director research to govern the research of the entire Punjab University, 78 departments and 188 affiliated colleges. So all these success to motivate, inspire, and the younger lot. कि लाइफ में बहुत कुछ है, करियर में बहुत कुछ है, फार्मेसी में बहुत कुछ है अगर आप चाहें तो. So yeah, I got this German invitation multiple times. Made the member of the Phospholipid Research Board in Heidelberg, Germany. So it was a great recognition, international recognition. First time I crossed the boundary of the country border, not to gain any expertise, to give expertise rather. So it's a different way to look at it. Of your recognition or success. So but still it goes on. बहुत कुछ अभी करना है, अभी कुछ हुआ नहीं है. It is just a glimpse of the things that one can do once if one aspire to. So resistance, cancer, mental, virus, diabetes, सभी कुछ बहुत कुछ आपके आगे एक चैलेंज है. मूव उनका जो है ना, बोलते हैं ना कि मूव आपके जो डिफिकल्टीज हैं जो आपकी बहुत ज्यादा हैं, यू हैव टू डू अ लॉट. तो how to make it possible, how to find that Sanjeevani. Now it starts the thinking and thinking and thinking. आज हमें सोचना ही होगा कि हम इस काम को अंजाम कैसे दें, हम कैसे वो मेडिसिन लाएं। और आज जहां हम खड़े हैं, we are celebrating, we are that India is number 2 or number 3 in the pharma, but still people are dying with the simple things, not simple things, but the diseases like tuberculosis and dengue, malaria, corona we have seen. So humanity is suffering actually. Whatever success in terms of money or in terms of whatever the medicines we are producing, but there is a long way to go. There is a the मंजिल अभी मुझे काफी दूर ही लगती है। So पराक्रम काफी बड़ा. It is like just like a bringing Ganga down, and it needs Bhagirathi Pratna. Bhagirath, our mythology, we appreciate the efforts, that entrepreneurial skills of the Bhagirath. So we need that Sanjeevani. We have to work hard. What is the philosophy it comes into the picture is, just think of a Newton, that he was the one to ask this question, that why this apple is dropping down. Everybody was seeing, but his mind was different. In its relaxed state, he could, his mind could only contemplate the principle of the gravity. This is the wonder of the questioning, wonder of the question. Question बहुत important है. अगर हम प्रश्न नहीं करते हैं और सही प्रश्न नहीं करते हैं तो वहीं खड़े हैं जहां पे खड़े थे. ये प्रश्न करना बहुत ही important है, और बिना प्रश्न के उत्तर आएगा कैसे? सबसे बड़ी बात तो यही है. तो हमें क्वेश्चंस ही नहीं पता है तो हम फिर कैसे रिसर्च करेंगे? So for having questions, you have to apply mind, and to apply mind, at least you should have mind. One should have mind. What mind? A searching mind, quest. So all these philosophical questions we look, and I got this as a very role modeling history, which encourages me, say, my own inspiration. I derive this divine inspiration, we call it, through this couplet, where Charak is to be proved as a father figure of the nanoscience, nanomedicine, through this couplet that we had in Charak Sanhita. And what is the meaning of this is that कि अल्पस्यापि मतलब बहुत छोटी मात्रा में भी drug का importance, it takes quantum leap when it is mixed with the right components and proper conditions and given in the proper time.
These are for that assay unknown sample. I will give you. I'm giving you. Wait. This is for the assay. कहाँ? नहीं, over here you are supposed to perform. FT के लिए मैं बोल देती हूँ. DT सामने है, TISO भी सामने है.
Am I audible?
Yes, sir, you are audible.
Okay. So this couplet slok by Charak is an eye opener, that how he had the idea of nanomedicine that we are celebrating today in our pharma sciences or formulation sciences. So, but and those times the discipline of medicine was run through Vaidya, and they were two in one, pharmacist and doctor both together in one mind. So this is the क्या कहना चाहिए tragedy of the medicine field today in the modern medicine, that we have kept two minds far apart. One is a doctor, another is a pharmacist, and even we are not meeting with them and not having the exchange of the knowledge, while our Vaidyas were actually having the command on the two. So they were able to had that impact, and this couplet actually is the proof of the concept. But we see only the picture which is very topical, which is very cursory in this couplet. We see Charak as a Vaidya. In my opinion, Charak was not a actually he did not go to the school, the Gurukul, to search medicine, but they were the true seekers. They were searching for the actual questions, actual knowledge, let's say, about the life. Life was considered as school. So Gurukul, those people were actually oriented towards, totally focused on spirituality. Unfortunately, we have destroyed that a fundamental basis of our education system. Earlier in those times, all our Aryabhatta, Charak, Dhanvantri, they were actually seeking the knowledge, true knowledge, and out of their hobby and interest, they become physicists, Aryabhatta, and the pharmacists and the doctors and whatnot. So, and the fundamental, the top operating thing in their search was to unfold their own mystery. Who am I? Where I have to go? These questions were there, the fundamental questions of the life. So because of that degradation, we are losing a lot. Our value system is also gone. Because we are only money oriented. On those times, Vaidyas were not charging, they were not looking money, they were looking for the to help people out of their compassion. And there was a only the service in the mind, helping the humanity was in the mind. Now we are, our pharma companies and all, they look only for money. How much money I will return, only I will make that drug, and the margin kitna hai, or you see these marketing people, they earn more than anybody else because how they are selling the product by hook and crook, and they pocketing the money. So where is the humanity? Where is the human service in that mad rush of the money and materialistic world? We have lost. That's why we have this imbalance and disharmony all over the places, and we are actually running haywire. So this is a philosophical question where the spirituality is makes a lot, and this because of the pursuits of the spiritual pursuits, we had a wonderful discoveries in the science. So even if you look, this nano medicine talks about the subtleness, sukshmata. So I have got these two examples that one is of a crane that if you have to pick up the needle, you cannot pick up by the crane. आपको जो सूक्ष्म चीज है उसको पकड़ने के लिए आपको सूक्ष्म इंस्ट्रूमेंट की जरूरत है. तो यही बात है कि हम लोग बहुत भारी भरकम चीजें सोचते हैं. सिंपलिसिटी वी हैव लॉस्ट. एंड महात्मा गांधी सेज की पोटेंसी ऑफ द थॉट लाई इन सटलनेस. अगर आपका विचार जितना सूक्ष्म होगा, मतलब आपकी जो सूक्ष्म विचार का मतलब क्या है, माने हाउ प्लेन, हाउ प्योर यू आर, प्योरिटी ऑफ इंटेंशन, and the ego, and how spiritually actually spiritual inclination and your these things are very important in personality, to acquire that leadership, that excellence, that ability to attract the people, collaborate the people. Your personality is very, very important. And getting out of your self-centeredness, selfishness, they matter a lot. In long run, you cannot, they pay, uh, and nobody is fool in this world actually. Everybody understand कि आपके intentions क्या हैं. So all these the philosophical dimensions, we are not working. We have actually become very, very aloof of these systems, taking them as a Dakiyanusi way, or these things. It is not so. Spirituality has a bigger role to play in future, and still it is played to role. Without spirituality, we cannot survive actually. We cannot survive. But to talk of the searching the medicine, searching the solutions, pharma solutions. So here in this direction, I got my hanged. Some problem, it is hanged. Can I restart because it is hanged? Is it visible now?
Yes, sir, it's visible.
Okay. So search for the master key. What is the actual solution? I'm not posing only problems, but I'm looking for the solution. हमारा ये नहीं है कि हमने आपको प्रॉब्लम बता दी तो आप प्रॉब्लम को आप सॉल्व कैसे करें. Search for the master. There is master key. There is nothing secret. It's an open secret. First, we have to think of our own personality, and in personality, what matters most is the mind. So to manage the mind, to make it more powerful, more effective, capacity building. When we talk of in research, mind is the first thing. So meditation is the only technique where we can actually find the state of the mind, that is called the meditative state of mind, the cool mind, the contemplative mind. Look to Dhanvantri and Shushruta and Charak. They all had a lot of tapasya and lot of dedication to the practice, abhyas, and the lot of efforts in their self-development, the self-exploration, and they became the great professionals, researchers and all. So meditation is the mantra. Without meditation, we cannot moderate, manage our mind because there is an inner conflict and inner disharmony. So without having a recourse to any spiritual way or spiritual thinking, and using these such practices like meditation, but we have been, as it's a very well correlated that when we meditate, then our consciousness that is a three-dimensional, it turns out to be. So correlating the data from left to right and from top to bottom, from three-dimensional, it is very important to have the multi-dimensional thinking. And only a cool and calm mind contemplate. Thinking in a unidimensional layer, it creates only a tunnel vision. You are not able to see the around. यहाँ से वहाँ नहीं दिखता, सीधा सीधा दिखता है. तो our liver doctor, they do not see the other side of the coin. So when I ask the Dharma people to look towards the liver to treat the skin, they remain aloof because they're only looking to the skin, while the liver plays a very important role in skin disease, according to the Ayurvedic system. So we need a mind which can actually expand like a balloon, and this ballooning is only possible when you work on your inner persona, where ego is down and your mind is open. So there is a rheology of the mind where the turbulence, a lot of turbulence in the mind. How can you be a contemplative? How can you be a researcher? How can you be leader? How can you be a good collaborator? How could you be a good human being? What to talk of all these things, you cannot become even a good family man. So you need a cool mind. You are still still water runs in the river, has got a very high current, high speed with the Reynolds number reducing down. And so this is the rheological aspects of your mental flow, which is very, very important. It goes very well to understand the importance of this dimension. So what we have already, your college, I'm thankful to Dr. Gaurav, they have understood the value of this dimension. They are having Heartful Campus programs on their campus. So these are these 4 simple practices that my organization, Heartfulness Institute, Khana Santi Vanam, they offer only 50 to 60 minutes around. They will teach you free of cost meditation and how to relax your body and how to clean your in the evening all the impressions that you do not want to carry. It's a very effective process, and your inner connect in the while going to bed, you connect yourself with you yourself inside. So there is an inner connection that matters. These dimensions are very, very important to enjoy the life, to enjoy the profession. And let's meditate. Kind of a Hearts app is available. You can note it down, and you can find the trainer all over the world, 160 countries around. We are our presence of the 14,000, 16,000 heartfulness trainer, like a big people, and in a very humble way. So people are doing it, and we are running this program of the youth empowerment, how to correct, how to correlate with your time management with the spiritual concepts, how you become the effective leader, multitasking, managing the stress, your skills, communication skills. Very interesting when you communicate. We often miss the use of the heart. So heartful communication, we have wonderful faculty. How to develop your willpower, how to, because this is not, these are the things which are not available on any Flipkart or Amazon. You have to develop, and you should understand that elements, the fundamental elements, how to improve your ability to take that will, sankalpa. So इमोशनल इंटेलिजेंस, बहुत अच्छे अच्छे टॉपिक्स लेके ऑनलाइन के जमाने में we had the faculty from across the country and beyond. So we have this programs running on campus Punjab University. All are teachers. You can look forward this website पे ये available है Heartful Campus program. आप भी अपने यहाँ करा सकते हैं, कहीं से भी आप करा सकते हैं. Our team in every state are very active all over the country. So many IIMs and IITs, they are doing it. We got the award for this work. More than any Rashtrapati award, we cherish this movement to receive this award at Kanasanti Vanam, where 1 lakh people can meditate together, inaugurated by our Rashtrapati ji. He himself practicing heartfulness meditation technique and trainer also. So he came and inaugurated this recently. This Kana Shanti Vanam city is in the limelight, winning the awards for the huge tree plantation and the silent change maker, this guide of this heartfulness, Kamlesh D. Patel, Daji, very popular. And he himself is a pharmacy, LM College product, LM College alumnus. Then he went abroad and set up became the very successful entrepreneur, putting up the pharmacy chains across the Newark and California. And he now for last 10 years serving this for the cause and making many projects there in the Kana Shanti near Hyderabad. So these they were the award recipients from the institutes like IIM, IITs, where we got this award in 2000, just before the Corona crisis. We had this fortune to receive this award. We celebrated my team, where the faces are changing. This is a very old picture, but and we have a good inner company, the Professor Bhoop and all other professors on campus. We have good association, very trustful associations, which I keep on enjoying the relationship, trustworthy relationship. So I salute my alma mater, Sagar Vishwavidyalaya, salute my Guruji, Professor V.K. Dikshit, who actually taught how to be friendly, and Professor Vyas, how to be daring, how to do so. Your alma mater is always valued, and I have a very strong feeling that whatever I am today is because of this. We got a recent success with this new product which the country launched, this IPCA launched, and this recently I'm very happy to share that when I received a call from my Guruji, Dr. Kamlesh D. Patel, Daji, that it has shown a wonderful effect on a different application. So it was just like for me that मैंने pharmacy पढ़ी ही इसीलिए थी शायद, जीवन pharmacy तो सफल हुई, जीवन भी सफल हुआ. तो ये बहुत ही अनहोनी सी चीज थी, यानी जिसके मैंने सपने कभी देखे नहीं थे कि I will receive the call from my spiritual master, and he will appreciate me to that extent, and really now I don't have any regret losing my medical education, though I was so competent and qualified to get the admission, but now I'm thankful to pharmacy. So we had this recent tech transfer at Baddi, one company, our team recently, only last month we went there and got another product scaled up, Foliflex gel. So that company is likely to launch in the coming year. So finally, a formulator will give you some formula for the medicine, formula for the liposome technology, and formula for the success, especially for the youngsters. That in this formula recipe, thankful to one of my students, she took this formula and made it so impactful. हमारा जिंदगी को हमें एक स्कूल की तरह लेना चाहिए और उसमें हमारा लक्ष्य हमें डिसाइड करना चाहिए और सिर्फ उसी तरफ देखना चाहिए. ध्रुव जैसी प्रतिज्ञा, ध्रुव एक की बहुत अच्छी कहानी, जो हिंदी बेल्ट में है, ध्रुव की कहानी सब जानते हैं. बहुत छोटा सा बच्चा जब उसने मन में ठान ली कि मुझे ईश्वर की गोद में बैठना है, तो वो करके ही रहा. So for spirituality, to gain that knowledge, you need not to be Buddha, because aging will give you only the chance to get your blood pressure measured. आप सिर्फ blood pressure ही ना पाएंगे, पर बहुत पराक्रम करना होता है, इसलिए आपको ये जवानी का ही काम है. You need a youthful energy, vigor and vigor to chase the higher goal. So भागीरथ जैसा प्रयत्न होना चाहिए. When Bhagirath can bring the Ganga down, why can't we bring a cancer medicine here? And the most important thing is surrender and egolessness, where the bhakti devotion actually means that egolessness. Whatever, if we host ego, then it becomes very difficult to your presence in the company, in the society will become actually nuisance. So how a egoistic person can be collaborate, it is very difficult to think of collaboration with the, even so he may be Einstein, he may be Newton, but if he is hosting that ego, people will run away from him and he will not be a good leader, not be listening. So these devotionals and state of surrender, meditative mind, spiritual dimensions, they're very important for personality development and to become effective and making the success. When we talk collective success, so India can gain its status Bharat Mahan if we regain, we rethink on these lines. This fortunately enough, that new education policy started talking about all these value system, our roots. But we need people, we need youngsters, energy to come forward and give it an open mind and think of it and actually looking beyond. It means, so I am again thankful to this Vidya Dadati Vinyam, means Vidya ke aane se vinamrata aani chahiye. Humility is the ultimate thing. So if we are getting the education, we should not be high-headedness, but a humility. So final acknowledgement goes to my Guruji, my master, Kamleshji Patel. Incidentally, he's also pharmacist, as I told you, and main of formulation only.
I am contesting.
So this is the final thing from my side. I am thankful, and I hope there is something which I could get across into. I hope.
Thank you very much, sir. Sir, we are also conducting the heartfulness sessions in our college also, and they are very calming and they are good for the students from our side.
Yes, I know,
Sir,
because,
now I would like to request Dr. Ashwin Dhingra to give vote of thanks to sir. Please, sir.
Thanks, Dr. Priyanka. Good morning, all of you. With due respect, I, on behalf of Guru Gobind Singh College of Pharmacy, would like to express my sincere gratitude to you, sir. You are the ocean of knowledge, and we are so lucky to have a resource person like you for participating in this 6-day online refresher program. During today's session on liposomal technology, you have provided a deep insight about the topic. Thank you so much, sir, for sharing your time and experience with us. I hope that you will continue the interaction and continuous involvement with us, sir. Thank you, sir.
thank you, Dhingra ji.
Sir, please accept my sincere appreciation for the outstanding presentation you made today, amalgamated with the need of the hour and your experience, sir. And your slide you showed to us, sir, was nothing short of incredibility, information, and above all, they are interesting to listen to you always. And you also informed us, but also inspired and motivated us. You tell us how a teacher first, how he will learn, and then how he will earn from that learning. And I want to mention over here, sir, that on your footstep, Guru Gobind Singh College is also moving on that step. Our two patent on novel drug system is going to be published this year also, sir. And two big giants of herbals, they are ready for the technical transfer of our products, sir. And in this way, all the our colleagues who are listening to you, they can make their path and they can learn or earn from industry also by following your footsteps, sir. Thank you very much, sir, for giving us time. You were sharing your experience and knowledge with us, sir. Thank you very much, sir.
Thank you, Dhingraji, and my heartfelt thanks to Dr. Gaurav, and I'm especially thankful for your adopting this Heartful Campus program. And this is the key. This is the key to success, in my understanding. There is no limit. There is no limit for the potentiation. Now we should not regret coming to the pharmacy profession. We have a lot. We have a lot. Thank you so much.
Thank you very much, sir.
Excuse me, sir. Sir, there is one question in the chat box from the participant side that how we can increase the stability and compatibility of nanoparticles.
Stability and compatibility. It's a very general question that having stability and compatibility in if one is in pharmaceutics and formulation science, one should know the value of the materials, the value of the method. So material, method, and your mind is very, very important. 3M: mind, material, and method. You are there, you are playing. Every baniya knows, every businessman knows how to do the business. Your business is to select the material. Your business is to select the method and to make face the challenges. What stability? Stability of the API, stability of the carrier system, and the compatibility vis-a-vis that, what applications. There is a whole lot of things which one has to address. So it is like you are in a you are on the ground. You are on the ground, and you have to play your game, and you have to select your shots. You have to select your things. It's like playing, and if you sharpen your mind, if you aspire, aspiration is the key. आपका कितनी आपकी इच्छा शक्ति है, कितना आपका इंटरेस्ट है. एक्चुअली इंटरेस्ट सबसे बड़ी चीज है. तो हम ये कहें कि लाइपोसोम बना दो या ये बना दो, वो कर दो, ये सब सेकेंडरी चीजें हैं. आपकी अगर इच्छा है और इच्छा शक्ति है, इंटरेस्ट है, तो जो गोल्ड मेडल जीतने वाला जानता है, जिसको गोल्ड मेडल जीतना है या जिसको एवरेस्ट पे चढ़ना है, उसे जानता है कि उसकी तैयारी कैसे होनी चाहिए. सो कमांडो ट्रेनिंग, स्पिरिचुअल कमांडो, एंड वी नीड अ कमांडो अप्रोच. तो जब आपकी चाहत होती है तो सभी चीजें हो जाती हैं, और हमारे पास में एम्फोटेरेसिन, लाइपोसोमल बी जैसे सॉल्यूशंस आ जाते हैं. So there is no one go answer for your question, but definitely yes, there is a you need have a thinking brain, meditative brain. If you aspire, you will get. Nothing can stop. कोई किसी को रोक नहीं सकता है.
Thank you, sir.
Is it okay?
Yes.
Thank you, sir. Thank you very much. Once again.
एक क्वेश्चन और है यहाँ पे ना सर. आई एम यूजिंग ओलिक एसिड फॉर फॉर्मुलेशन ऑफ यूफाजोम्स. We are not able to get film. We tried all method, published inside diet. Sir, can you say? देखो ओलिक एसिड क्या ऑयली है ना. तो अब ऑयली चीज की फिल्म कैसे बनाओगे आप? ये तो है ना प्रॉब्लम है. ऑयली चीज को आपको किसी ऐसे सिस्टम में डालना होगा जो इसको स्प्रेड करा दे. So how can you think of a oleic acid to form a film? No. Alongside of the oleic acid, if you use something like phospholipid or a span, and to which are film former, so you think of forming a film with the help of the composition of the right kind of a selection of the lipids. So you can form the film like in iosomes. तो जब तक स्पेन के साथ हम कोलेस्ट्रॉल नहीं डालते हैं कैसे बनती है फिल्म? तो ओलिक एसिड की तो फिल्म बनेगी नहीं, कभी नहीं बनेगी। तो ओलिक एसिड के साथ में कंपनी चाहिए। जो पेपर जो छापते हैं ना वो लिखते नहीं है सारी चीजें, वो सिर्फ उसके लिए होती है, पब्लिकेशंस के लिए होती है। दे कैन नॉट, दे डू नॉट राइट ईच एंड एवरीथिंग actually, either in the paper or the patent also. So फिल्म बनाना है आपको तो आपको कुछ मेहनत करनी पड़ेगी कंपनी, और सोचना भी पड़ेगा। ऐसा नहीं है, theoretically you should be very sound. Is it okay?
Yes.
Phosphatidylcholine is very costly for me. What are the alternative? वो आप अलग से आओ मेरे पास बात करने। कॉस्टली तो है, दुनिया में सभी कुछ कॉस्टली है।
Okay, thank you, sir.
Okay, thank you, madam. I wish you the
Yes.
best for you and the college. Can I leave now?
Yes, sir.
Okay. So hope to meet you soon. No.
Thank you, sir.
You are welcome to Chandigarh. No? All.
Thank you, sir. Wish you all welcome.
Thank you. Thank you.
अगला किसका है सर? ओके, ठीक है।
हो गया नेक्स्ट
आगे
उस पे चेंज कर दो
नेट चेंज कर लो पहले अपना।
कहाँ रविंदर का है?
रविंदर चला गया।
यार तुम जाने क्यों देते हो? चलो उसका फोन लेके अपने पास रख लो।
ये बुलाएंगे कैसे?
ये हमारी आवाज
वही आ जाए।
Yeah, good morning. Can you see my screen, my presentation? Hello. Dr. Kumar? Is my screen visible? Dr. Kumar? Can you see my screen?
Yes, sir. Screen is visible.
Okay, can I start? Madam, can I start? Excuse me. Can I start my presentation? Yeah, can I start my presentation?
Rahul sir, just give us a 2 minute. Actually, there is some connection problem in internet.
Okay.
Just give us 2 minutes, sir.
Sure.
Just 2 minutes. Sir. We are. I'm really sorry.
So sorry for the delay.
No problem.
Sir, now I would like to introduce our next speaker of the day, Dr. Rahul K. Verma, Institute of Nanoscience and Technology, Mohali. He is recipient of CDRA Incentive Award and 2 times recipient of BRG France Travel Award. Dr. Verma has published 55 research papers and 11 book sections in highly reputed international journals and books respectively. His current research interests include engineered aerosol biomaterials for controlled delivery of drugs, peptides and vaccines, nanoscale manipulation of multiphase polymeric materials, bio-inspired materials, nanoparticles, etc. He is expert in formulation development using spray drying, spray freeze technology, supercritical fluid technology, and microfluidizer. In addition, he has also conducted research in preclinical pharmacokinetic, therapeutic, and toxicological evaluation in cell lines and animal models. I welcome you, sir.
Yeah, thank you. Thank you.
Sir, I request you to please take over.
Yes. Thank you, madam, for the nice introduction. Good morning, friends. As mentioned, I am Dr. Rahul Verma, working as Scientist E at Institute of Nanoscience and Technology, which is located in the Mohali, Punjab. I would like to thank the organizer for inviting me for AICTE-ISTE sponsored induction program. So since my institution is quite new, so I would like to give brief introduction of my organization. So AICTE is an autonomous institute of Department of Science and Technology, Government of India, which is under umbrella of the Nano Mission. Under guidance of the Bharat Ratna Professor C.N.R. Rao, the institute was established by DST to boost the research and development in the nanoscience and technology in India. And I want to mention that this is only institute which is completely dedicated to nanotechnology in India. And this institute has 3 divisions that are energy and environment, quantum materials, chemical biology unit. And our aim is to generate the technologies and the devices directed toward the national priorities, especially in the sector of healthcare, agriculture, defense, energy, and environment. So according to the Nature Index 2021, it holds the rank of 17 in chemical science groups among the top institutions in India. It is just after the IIT Delhi. Now coming to the presentation. My lab is basically working in the area of the pharmaceutical nanotechnology and the drug delivery systems, where we are involved in design, development, and evaluation of different modalities of the drug delivery system like nanoparticles, microparticles, liposomes, hydrogels, for the diseases like tuberculosis, lung cancer, cold injury, and rheumatoid arthritis. The inhalation is one of our prime research area. So in this presentation, I will give the brief overview of the experimental methodologies to design and evaluate the inhalation drug delivery systems. So this lecture will include inhalation drug delivery, lung as a portal for drug delivery, particle behavior in lung airways, factor affecting the inhalation delivery, techniques of making particulate matter in for lung delivery, evaluation methods, physiochemical characterization, dose uniformity tests, aerodynamic properties, uh, drug administration systems, deposition mechanisms, clearance of inhalation particles, factor affecting the inhalation delivery, and the techniques for pulmonary drug delivery like MDI, DPI, and nebulizers. So the delivery of the drug directly to the respiratory tract via mouth, either by mouth or nose, is called inhalation delivery. It is important delivery route for wide range of medications because it has ability to access the wide vast research surface area of the lung and target a specific location along the way. So in the recent years, it gained much interest of research as it can be used as local as well as the systemic for the systemic diseases. Uh, in the local delivery system, it is used for the management of the respiratory diseases like asthma, COPD, tuberculosis, pneumonia, cystic fibrosis, respiratory infection, etc. And in case of the systemic drug delivery, it can be used for the diabetes and heart diseases. The pulmonary drug delivery have significant advantage over the conventional delivery systems. Like it provides non-invasive needle-free mode of administration, require low dose. It has quite quick onset of action and evade first-pass metabolism of liver and cause least drug degradation. And there are few limitations which are associated with the inhalation systems, like difficulty in producing optimum aerodynamic parameters. Some drug may produce the irritation or toxicity, and some drug may be retained in the lungs, and clearance of the drug can be the difficult. Yeah, before going to the actual drug delivery system, the drug delivery by the pulmonary route requires a deep understanding of the complicated anatomy and the physiology of the airways. The geometry of the airways influences the deposition and the clearance of the delivery at the particular location of the respiratory tract. And the respiratory system consists of two regions, the upper and the lower respiratory tract. The upper tract includes the nasal cavity, nasopharynx, oropharynx, and larynx, while the lower respiratory tract consists of conducting airways that include the trachea, bronchi, and bronchioles. The respiratory region consists of the respiratory bronchioles, alveolar duct, and alveolar sacs. The large surface area of the lung, good blood supply, and the high permeation of the lung makes it ideal portal organ for the uh, drug delivery. So depending on their size and the shape, the particle can be deposited and transported in the respiratory tract by means of three mechanisms that are inertial impaction, gravitational sedimentation, and Brownian diffusion. In the inertial impaction, large particles above 5 micron undergo the inertial impaction and settle in the upper airways. And these particles unable to follow the air stream, impacting the airways walls or this respiratory tract walls. And as the branching continues, an air stream and impaction decreases. The gravitational sedimentation occur the gravitational sedimentation occur for the particles ranging from 1 to 5 micron, which settles under gravity and follows the airflow and settle in the bronchi and bronchioles. Here the airflow is slow, hence the resistance time is more. The smaller particles, which are from 0.5 micron to 1 micron or in the nano size range, undergoes the random Brownian motion and deposit into the lung wall. And diffusion is the most important mechanism for the deposition of in the small airways and the alveoli, and it occurs mainly in the nanoparticulate system or submicron systems, which usually goes to the alveolar region of the lung. Yeah, there are various techniques for making the particulate matter for the lung delivery. The spray drying is the most convenient technique to generate the particle for the inhalation delivery. In this method, dry powder or uh, or the particles are produced from the liquid or slurry by rapidly drying it with the hot gas. In this process, the feed solution is pumped into the nozzle where it is atomized by the nozzle gas. The atomized solution is then dried by the preheated drying gas in the chamber to remove the water moisture from the system and thus forming the dry particle. And these particles get collected into the cyclone collector. And there are two type of spray dryers which are available in the market. One is the conventional spray dryer, uh, which produce the particles of micron size from 2 to 100 micron, which are generated by the conventional spray dryer, which uses the axial nozzle. Which uses the axial nozzle. The conventional spray dryer produces the particle size of 2 to 100 micron, and here the nozzle is of axial type. Where the other type is the nano spray dryer, where the particle in some micron size or nano size are produced. Here we can produce 200 to 2 micron size particles. Here the nozzle is the piezoelectric nozzle, which produce the strong vibrations to form the fine droplets which make the nanoparticles. So application of the spray dryer can be the development of the inhalation drug delivery, inhaled drug from the dry powder inhaler, nano and micron encapsulation of the liposomes, encapsulation of a nanoparticle with high bioavailability, and the porous drug carrier of nanoparticle suspension. And it can also used for the development of excipient for the controlled drug release like trehalose, mannitol, lactose, HPMCs. The, the another important technique for the generation of the inhalable particle is the spray freeze drying technique. It involves the spraying of the drug solution into the liquid nitrogen as a freezing medium, followed by the lyophilization. Here, in this method produces light and porous particles with high fine particle fraction with improved aerosol performance. Here the nozzle of the spray dryer or atomizer nozzle is used, which generate droplets which get instantly freezed into the liquid nitrogen and produce the particles, which are then lyophilized to produce the hard particles. This this system is basically used for the thermolabile proteins and peptide substances such as insulin and plasmid DNA that can also be formulated into dry powder inhalation product. The other useful technique is the supercritical fluid technology. It is an expensive and the less used technique for the generation of inhalable particles. It depends upon the controlled crystallization of the drug from the dispersion or in the supercritical fluid. The supercritical fluid can be the liquid carbon dioxide, and here the drug and the carrier molecule. Here the drug and the polymer molecule are dissolved or dispersed in the supercritical solvent, that is CO2 carbon dioxide, at the critical temperature. and the pressure conditions, critical pressure, temperature and pressure conditions. And then it is sprayed through the nozzle into the chamber where it is expanded, which results the loss of the solvent of the supercritical fluid and which precipitates the drug particles as a drug particles in a fine particle form, dry powder form. And this method is also used for the thermolabile substance like protein, peptides, and DNA. The other common techniques are the solvent precipitation method and double emulsion solvent evaporation technique. These are the common techniques used for the generation for nanoparticles and microparticles in most of the labs. In the solvent precipitation method, which involves the sonocrystallization and microprecipitation, the crystalline drug with a narrow size distribution is prepared by direct control of crystallization. Here, the inhalable particles are produced by rapid precipitation of the aqueous solution using the anti-solvent systems. And the other technique, that is double emulsion solvent evaporation method, involves the preparation of the emulsion with the subsequent removal of the oil phase from the through the evaporation. Here, the organic solvent diffuses out of the polymer phase into the aqueous phase and then it get evaporated, forming the drug-loaded polymeric nanoparticles. These two techniques are very simple and being used in most of the labs which are working in the pharmaceutics or drug delivery systems. There are various evaluation methods for the inhalation drug delivery systems. For characterization of the inhalation delivery system, we do the physiochemical characterization, aerodynamic characterization, in vitro evaluation, and in vivo evaluation. Physiochemical characterization involves the particle size and charge evaluation, morphology, solid state characteristic that involves the thermal analysis, X-ray diffraction, XRD, spectroscopic analysis like Raman, NMR, IR analysis. Then quantitative surface analysis, which is done by BET and BGH. And we also do the interparticle force measurements. And in case of the aerodynamic characterization, which involves the flow properties, that involve the car index, Hausen index, those uniformity test. The most important is the mass median aerodynamic diameter evaluation in the inhalation systems. Then the fine particle fraction, geometrical standard deviation. The last three, the mass median aerodynamic diameter, fine particle fraction, and the geometrical standard deviation are the most important parameters for the inhalation system. The in vitro evaluation, we do the cell culture, the uptake studies, and toxicity studies. And in vivo evaluation methods, we do the drug administration studies, which involves the whole body exposure system or nose only exposure system or intranasal exposure system. And we also do drug deposition studies and the pharmacokinetic studies in the animals to evaluate the inhalation systems. In the physiochemical characterization, the particle size and the zeta potential are the crucial factor, which decides the efficacy and the stability of the drug delivery systems. The particle size dictates the penetration of particle into the lungs or the systemic delivery. And the zeta potential help in the stability management of the system. The particle size less than 5 micron has high possibility to deposit into the lungs. And the particle which has size lesser than 2 micron has probability to reach into the alveolar region of the lung. And the particle size having less than 1 micron are usually get exhaled during the breathing process. And this is not the appropriate for the inhalation system. The particles below the 1 micron are usually not in the dry form are not suitable for the inhalation because they get exhaled. And we do the size analysis by two methods. One is the zeta sizer machine and other is the master sizer. The zeta sizer is used for the charge measurement and the particle size measurement of the nanoparticle size or submicron size, while the master sizer works on the laser diffraction method, which is used for the micron size particle measurements. The morphology of the particles are generally evaluated by three methods, like transmission electron microscopy, scanning electron microscopy, atomic force microscopy. In transmission electron microscopy, particle shape, size, structure, and arrangement can be identified by the interpretation of morphological data of by the TEM analysis. In case of the scanning electron microscopy, which is used to evaluate the particle size and surface morphology of the particle. And in the atomic force microscopy, the interparticular forces can also be measured with the surface energy and the morphology of the particle. Here, additional advantage in the AFM, there is additional advantage to measure the interparticular forces and the surface energy with the particle shape. The another parameter which is very important for the inhalation evaluation of the inhalation drug delivery system, that is powder flow characteristic. There are several parameters that dictates the flow properties of the powder, that are angle of repose, bulk density, tab density, porosity, surface area, car compressibility index, and Hausner ratio. The angle of repose is the angle that the side of the conical heap of powder makes with the horizontal plane. This represents the flow of the dry powder. The bulk density of a material is the ratio of the mass and the volume of untapped powder sample. This is the bulk density, which includes the interparticulate void volume also. With the and the tap density is that the tap density is obtained by the mechanically tapping the graduated cylinder, glass cylinder containing the sample until the further volume gets changed. So major difference is that in the bulk density it includes the interparticular void volume, but in tap density we do not calculate we do not include the interparticular void volume in the tap density. The porosity and the surface area is usually evaluated in our labs by BET surface analysis method and the BGH pore size and volume analysis method. The Car's compressibility index and the Hausner ratio, which indicate the flow properties of the powder. These parameters are calculated by the bulk and the tap density values. The car index, the car compressibility index value from 0 to 10 shows the excellent flow properties, while from 26 to 31 shows the poor flow properties. And we usually consider from 0 to 15 car index is appropriate for the inhalation systems. The intraparticular forces measure is also important parameter for the inhalation delivery systems. Here the adhesive forces or electrostatic forces between the carrier and the drug particles are crucial, as the dose deposition in the lung depend upon the separation of the particles from the carriers. And these forces are can be measured by the direct or indirect method. Here, atomic force microscopy colloid probe technique is generally used as a direct method for the measurement of this cohesive adhesive measurement or adhesive forces measurements. And in the indirect method, inverse gas chromatography method, centrifugation, and particle detachment rate measurement. These are the method used for the evaluation of the adhesive forces of the inhalation systems. The delivered dose uniformity is a critical quality attribute in determining the safety and quality efficacy of the inhalation drug products. And it is routinely measured by the equipment that is called DUSA, that is doses unit sampling apparatus. This machine is available in our lab, and we routinely use for the delivered dose uniformity testing. Here, the delivered dose is retained by the filter in the sample apparatus after the actuation of the test device. And the active drug from the retained dose is measured. And here, the simulation of the inhalation is brought about by drawing air through the sampling apparatus. And the amount of retained in the filter of this machine uh, is calculated as the delivered dose for the inhalation, and this dose will be available for the inhalation by the patient. The another pattern, another parameter is the spray pattern and the plume geometry characterization, which is equally important during the development. And this is also the regulatory requirement for the inhalation the nasal products. And this spray pattern data is also required for the release testing. And the shape, angle, and duration of the plume is highly influential in the uniform delivery and viability of the product. And ultimately, it is required for the optimum efficacy of the inhalation products. The plume geometry is characterized by the spray angle of the plume and the width of the plume. Here, the spray angle is measured from the vertex of the spray cone and the conical nozzle. And the plume width is the width of the plume at a given distance from the spray nozzle. These measurements are mainly done by the Proveris spray view machine, which incorporates the actuation and high speed imaging to obtain the spray pattern parameters. It measures the following parameters like plume orientation, cone angle, plume width, and plume length, and the distance from the nozzle. This gives the idea of the spray pattern of the uh, inhalation device. Yeah, the most important characterization parameter for the inhalation product is the aerodynamic factors. The aerodynamic factors influencing the pulmonary deposition includes the aerodynamic diameter that usually calculate the mass median aerodynamic diameter, fine particle fraction, and geometric standard deviation. Aerodynamic behavior of the airborne particle having irregular shape are expressed in terms of the diameter, which an idealized spherical particle known as aerodynamic diameter. This is an imaginary spherical particle that is conserved equivalent to the irregular particle. The MMAD is the mass, is the median of the distribution of the particle mass with respect to the aerodynamic diameter. And it is defined as the diameter at which the 50% of the particle by mass are larger and 50% are smaller than the stated diameter. And MMAD is the average size of the particle constituting the dose which reaches to the lung. Then the fine particle fraction is the fraction of the particle that can be achieved deposition in the lower respiratory tract, that is below 5 micron. It is usually below 5 micron, and it is the fraction of the particle that comes under potential deposition size range. And the fine particle gives the size range of the particle which can actually deposit into the lung from the bulk of the powder. And the geometrical standard deviation is the measure of the spread of the aerodynamic particle size distribution. The ideal aerodynamic characteristic for optimum deposition in the lung includes the particle size of 1 to 5 micron, If the particle size is 1 to 5 micron, it has high probability to get deposited into the lung. And the ideal mass median aerodynamic diameter should be 1 to 3 micron. And the fine particle fraction should be more than 40% to have the optimum drug deposition into the lung. And the standard deviation should be in the range from 1 to 2 micron to have the optimum deposition into the lung. The aerodynamic properties and the in vitro pulmonary deposition of the inhalable formulation is measured by using the cascade impactor. So in our lab, we are using next generation cascade impactor and Anderson cascade impactor to measure these aerodynamic properties of the inhalable products. In the cascade impactor have the 7 to 8 plates. You can see the plates over here arranged horizontally, which have different pore diameters. It has the different pore diameters. And these different plates of the cascade impactor mimics different parts of the respiratory tract. Like stage 1 mimics the nostril, stage 2 mimics the pharynx, stage 3 mimics the trachea, stage 4 bronchi, stage 5 terminal bronchi, and stage 6 and 7 mimics the alveolar region or alveoli region of the lungs. The inhalation device having the formulation is attached with the mouthpiece of the cascade impactor, and the actuation is given. And then the cascade impactor segregate the particles according to their aerodynamic size, and it gives the idea of the fraction of powder deposited in each part of the lung. It gives an idea which fraction of the powder will deposit into which part of the respiratory tract. So after this data, we can calculate the mass median aerodynamic diameter, fine particle fraction, and geometrical standard deviation using the generating the graph between the effective cutoff diameter, that is the pore size of these different plates of cascade impactor, and the cumulative percentage undersize, that is the deposition of the powder in the different the 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 mass of the particles that deposited in different plates. It is against the effective cutoff diameter and the mass of the powder deposit at each plate of the cascade impactor. This is the most important parameters to evaluate the inhalation system. So for the drug administration to the animals or the humans, for the experimental purpose, we use two types of the exposure systems. One is the whole body exposure system, and other is a nose only exposure system. In the whole body exposure system, which is a sealed plastic chamber or a steel chamber attached with the powder aerosol generator or nebulizer, and the animals are placed inside the chamber. Here a nozzle generate the aerosol, and it get inhaled by the these animals. But in comparison of with nose only exposure system, the whole body exposure adds the least stress than that of exposure material, and making more suitable for the long term and the repeated inhalation. But there are several there is still some concern about this exposure system, and because it can cause the contamination from the other routes other than the nostrils or the mouth, and has an effect on the particle deposition, and it can also have effect on the health of the animals. The nose only exposure systems are the systems in which the animals are restrained in the exposure chamber, and the aerosol in contact with only head and nose of the animals, and it get restrained with this chamber, and then actuation is given. and the available inhalable particles are inhaled by the these animals. The merit of a nose-only chamber are that the exposure concentration can be high, and there is less waste of the this formulation because the chamber is small, has a small volume, and it goes directly to the lungs from the nostril or mouth through the nostrils, and avoid the entry of the particle to the other routes, and it goes by only inhalation route of the administration. The amount of drug deposited in the lung and the its distribution are the key parameters in the evaluation of the formulation and the performance of device in the pharmaceutical aerosols. So the deposition of the lung can be measured by invasive or non-invasive methods in the experimental animals. The non-invasive techniques provide it gives in vivo pharmacokinetic parameters with respect to the time course of the drug administration. In the non-invasive technique, we use the single photon emission computed tomography, gamma scintillography, and positron emission topography. These are the imaging methods which can generate data of the deposition. This is mainly used in the animal imagers, which gives an idea how much drug is deposited in the lung or any other part of the body. And these imaging methods are successfully employed in the pulmonary drug delivery system and inhalation toxicity studies. In the invasive technique, we do the bronchoalveolar lavage, uh, which is the method that is employed for the insoluble inert particles when non-invasive methods are inadequate to measure the drug deposition. The bronchoalveolar lavage are usually conducted by the tracheotomy method to recover drug from the airways and the lungs. Here, the animals are anesthetized and the trachea is cannulated using cannula, and the lungs are lavaged using the chilled saline or any other solvent. So, and the lavage obtained is pooled, and then it is estimated for the particles or the drug present in the airways or in the lumen of the lung. There are different parameters which are essential for designing the effective inhalable formulation. These are the physiochemical properties like particle size, particle shape, charge density, solubility, mass, median aerodynamic diameter, geometric standard deviation, fine particle fraction. Then the geometry of respiratory influence of the disease state on the airway structure. Then the breathing pattern, whether it is mouth or nasal breathing. Like physiochemical factors influencing the pulmonary deposition include the particle shape, size, density, and the particle roughness. During aerosolization, the particles with the different shapes subject to different drag forces and terminal velocities, which affect the deposition in the lungs. Uh, in an in an study by Hassan et al., who evaluated the deposition pattern of particles of different shape in respiratory tract using the cascade impactor. They used the particle of almost similar size, but the size was same, but the shapes were different. They used the spherical, spheroids, pollen shaped, cube shaped, needle shaped particles. And after cascade impaction or the evaluation of the aerodynamic parameters, the results shown that the pollen shaped particles showed the highest deposition, followed by the spherical, then spheroids, then needle shaped, then cube shaped, and then large needle shaped showed the lowest deposition in the lungs. And here, particle density also plays an important role in the deposition. The study shows the lower the particle density, the better will be the flow properties and higher will be the deposition in the lungs. Another property that affects the deposition pattern is the roughness. If the particle is more rough, then there will be high degree of deposition into the lungs. They get entrapped into the lung because of their roughness. So that's why here the pollen shaped particles showed the high deposition as compared to the other spherical or spheroid particles, because they get deposited and they cannot be moved uh, by uh, by the macrophages or other systems, clearance systems. Now, coming to the clearance mechanism of the inhalation formulation, which depends upon the properties of the particles of the formulation. If the particle is soluble, then it get absorbed into the bloodstream after dissolution within the airways, lining fluid or cells, and the clearance occur within all regions of highly vascularized respiratory tract to the bloodstream. It goes directly to the bloodstream after absorption if the particle is um, water soluble. And but if it is insoluble, then it will be cleared by the lung by two mechanisms. If the particle is insoluble or polymeric, then it will be cleared by two mechanisms. One is the mucociliary clearance and other is the macrophage mediated clearance. There are numerous cilia present in the bronchial wall epithelium that beats in wave pattern and remove the particles, and this particle transported to the larynx and then swallowed by the GIT. So this is the one mechanism where it goes into the gastrointestinal tract and gets swallowed. And other mechanism is the macrophage mediated, where the particle are removed by the macrophages. It causes either degradation of the particle by the lysosomal enzymes or the transportation of particle to the lymphatics via macrophages. Here the macrophage take the particles, phagocytize the particles and take it to the lymphatic systems. Okay, there are different types of the nano carriers being designed and developed for the pulmonary applications. The most important is the drug-loaded polymeric nanoparticles. These nanoparticles either can be used as dry powder inhalation or dispersed liquid droplets dosage form in the meter dose inhaler and nebulizers. Drugs either encapsulated or bound on the surface of the nanoparticles. These are usually prepared by the nano spray drying, emulsion solvent evaporation method, milling or supercritical fluid technology. These polymeric nanoparticle based formulations are in different stages of research and development for the diseases like asthma, tuberculosis, cancer, hypertension, lung infection, and other diseases. The other type of formulation is solid lipid nanoparticles, which are submicron colloidal carriers, which possess the solid pore matrix and then solubilize the lipophilic molecules. These can either be used as a dry powder inhalation using the excipient like lactose or trehalose, or dispersed liquid droplet form, which is used in the meter dose and the nebulizers. And the method of preparation for these particles can be the high shear hot homogenization method, ultrasonication, double emulsion method, nano spray drying. And here also these formulation are in different stages for the asthma, COPD, uh, tuberculosis, cancer, hypertension, and infection diseases. The other formulation, very important formulation, is the liposomes, which are being used for the pulmonary delivery, and it has high market interest. And these vesicles entrap these lipid molecules. These lipid vesicles entrap the therapeutic molecules following the inhalation and localize drug effect in the pulmonary system for the prolonged period. These are mainly used for the meter dose inhalers and the nebulizers. These are less explored in DPIs like dry powder inhalation. These are less explored, but these are frequently used in meter dose inhalers and in the nebulizers. And the method for the preparation is the membrane extrusion liposomes, ethanol injection method, membrane hydration, sonication, and microfluoridation. These are also used for the respiratory diseases, for asthma, COPD, tuberculosis, cancer, hypertension, lung infection. And these are also used for diabetes, for for inhalation delivery of the insulin and the glucose-like peptide 1, which is also an anti-diabetic peptide. And these are also used for the siRNA gene delivery and the delivery of the protein and peptides via inhalation. And there are some hybrid kind of nano assemblies which are being used for the inhalation. In our lab, we developed this kind of formulation which are called hybrid nano assemblies or nano in microsystems, where these are nanoparticles which get aggregated into micron size, because the nano size get easily exhaled while the breathing process. So we develop the nano in microsystem or PNAP, that is the porous nanoparticle aggregates, which has the properties of the nanoparticles, but the size is of micron size, which can be easily inhaled and get entrapped into the lungs. So here the advantage is that this these has appropriate aerodynamic diameters, and they can have the appropriate deposition to the lungs, and after as they reach it to the lung, they get dispersed into the individual nanoparticles. So they can cover the more area as compared to the conventional microparticles. They get dispersed into nanoparticles into lungs and cover more area and will have the more efficacy as compared to the conventional microparticles we use. So these systems are developed in our labs only. And we also develop different kind of the micro particles for different applications. We develop the porous microspheres, highly porous microspheres, golf-like particles, then mucus penetrating particles, a different type of mucus penetrating particles, which are these mucus penetrating particles are more appropriate for the diseases like cystic fibrosis and then tuberculosis, and the diseases which forms high content of mucus in the lungs. These particles penetrate the mucus and reach to the target into the in the lungs. So we also developed this mucus penetrating inhalable particle for the TB therapeutics, where we generated polymeric PLGA particles and decorated with the N-acetylcysteine. And these are able to give the enhanced translocation through the mucus. And these have, we found that these particles have high mucus penetrating power, then anti-biofilm activity and anti-tubercular activity. And this was published in Journal of Controlled Release. I will skip this slide. The aerosol formulation are the stable dispersion and suspension for solid materials and the liquid droplets in the gaseous medium. There are three commonly used inhalation devices for the aerosol, clinical aerosols, that are dry powder inhalers, meter dose inhalers, and the nebulizers. The basic function of these three completely different devices is to generate the drug containing aerosol clouds that contain the high possible fraction of particles in the desired size range, which can be easily taken by the patient through inhalation. The DPI or dry powder inhalation contain the solid drug in the dry powder mix formulation. When the patient, this contains the solid drug in the dry powder mix that gets fluidized after the actuation of the device when the patient get inhaled. And DPI formulation is inhaled as a cloud of fine particles. The drug is either preloaded in the inhalation device or the filled in the hard capsule, hard gelatin capsule, or the foil blisters disc, which are loaded into device prior to use. These dry powder inhaler require the particle size of 3 to 5 micron uh, size, and to improve the flow properties of these particles, these are usually mixed with the excipients like the lactose or trehalose. And the advantage of this system is that DPI can deliver the high doses as compared to the MDI. This is the propellant free, so there is less or no toxicity, and there is less need of the patient coordination and less formulation problems. And these are mainly used for the asthma, COPD, systemic disorders like diabetes or lung cancer, and also neurological diseases. There are two type of inhalation. This dry powder inhaler, that unit dose powder inhalers. These devices are with contained capsule, which is placed in the holder, and the capsule is opened within the device, and powder get inhaled. This type of inhalers are available in the market, which are spinhaler, rotahaler, and aerohaler. In a multi-dose powder inhaler, the drug contained in a storage reservoir, which dispense a unit dosing at one actuation. This kind of system is available with the turbohaler and actuahaler and clickhaler, which are available in the market. Another system is the meter dose inhalers. In this, the drug is dissolved in the liquid propellant mixture with a metering valve, and they can be given in the form of solution and suspension. Here, the predetermined dose is released as a spray on the actuation of the metering valve. When released, the formulation undergoes the volume expansion and form the mixture of gas and liquid before discharge. The high-speed gas flows through it and break the fine this liquid into the fine spray droplets. Here, the particle size is 2 to 5 micron. Here, there is need of using suspension or suspending agent, which make these particles suspended in the liquid, and it delivered very precise. The advantage is that it delivered very precise amount of dose. It is small and convenient, and it is less expensive as compared to DPI and the nebulizers. It is quick in use, and it has capability to give the multi-dose more than 100 doses with one device. Other inhalation system is the nebulizer, which is widely used to aerosolize the drug solutions and the suspension of drug delivery to the respiratory tract. And this is particularly used for the treatment of the hospitalized patients. And it delivers the large amount of the drug solution and suspension. And it is frequently used for the drug that cannot be conveniently formulated or delivered through the MDI or DPI. There are two type of nebulizers. One is the jet nebulizer, and other is the ultrasonic nebulizer. Jet nebulizer uses the compressed gas that convert liquid into the spray, and the compressed air with the high velocity passes through the orifice, and it causes the liquid to draw up to feed pump from the fluid reservoir. Here these are mainly used for in the hospitals. And in case of the ultrasonic nebulizer, the energy for the atomization of the droplets is carried by the piezoelectric crystal vibrating at high frequency. And this ultrasonic intensities develop the droplet, which is inhaled by the patients. So with this slide, I will come to the conclusion that a number of factors affect the deposition of the aerosol in the lung. Aerosol particle plays important role in targeting lung region. Appropriate drug formulation and devices need to improve the efficacy and pulmonary drug delivery remain the preferred route for the administration of the various drugs. Thank you.
Thank you very much, sir. It was a very informative session, and I think you elaborated well about the inhalation drug delivery systems and the various formulations like nanoparticles, liposomes, and microparticles, their evaluation parameters, and technologies used for the preparation of the dosage forms. So, thank you very much, sir. As any of the participants, if you want to like to ask any of the questions. Participants, if you want to ask any of the questions, you can ask. You can unmute yourself. You can also write the chat box.
Yeah, I can see the chat box because you are not audible. The student is not audible.
Okay, okay. Okay, thank you very much, sir. Sir, as a token of appreciation, would like to present a certificate to you. It's on the screen.
Oh, thank you so much. Thank you so much.
Thanks. Thank you, sir, for a wonderful session. Okay, participants, we shall be meeting again at 2 o'clock for the second technical session. So, thank you very, very much, all of you.